Thanks everyone for joining another trainee rounds hosted by T-CAIREM. My name is Arman. I'm a current T-CAIREM education affiliate and also a PhD student. So I'll be hosting today's rounds. We have two excellent trainees presenting really exciting work. So we'll go ahead and just jump right into it. We'll hear from two trainees, first from 12 to 12:30, Suraj Bansal about ATLAS-AML, an automated bioinformatics pipeline for drug target characterization and discovery in acute myeloid leukemia, followed by from 12:30 to 1 o'clock, Sidrah Laldin presenting comparing innovative AI algorithms to assess echocardiographic videos for clinical modeling. Just a reminder, the presentations will be 20 minutes, followed by 10 minutes of Q&A. We have two adjudicators as well as myself who will begin the question periods, and then anyone can put questions in the chat, and we'll try our best to get to those within a 10-minute period for each presenter. So without further ado, we'll jump right in. The first presenter, as I mentioned, is Suraj Bansal, as a first-year medical student at the University of Ottawa and a researcher in Dr. John Dick's lab at the Princess Margaret Cancer Centre. He's an aspiring clinician scientist and is passionate about leveraging single-cell multiomics, machine learning, and experimental approaches to study disease heterogeneity and develop targeted therapies for acute leukemias. We look forward to hearing about ATLAS-AML. Suraj, please take it away.
All right, thank you so much for the introduction. I'm just going to share my screen. Are we all able to see this okay?
Yes.
Okay, excellent. Okay, so thank you again for the introduction. My name is Suraj. I just finished my first year of medical school at the University of Ottawa, and I'm also a student researcher in the John Dick lab at Princess Margaret Cancer Centre. And today I'm going to be talking to you guys about a pipeline called ATLAS-AML, which is an automated bioinformatics pipeline for drug target characterization and discovery in acute myeloid leukemia. And acute myeloid leukemia is just a really aggressive and heterogeneous blood cancer that has really poor survival. And to start this off, I want to begin by talking about the valley of death, or what I call drug discovery and development in acute myeloid leukemia. Because although there's a lot of really promising drugs that are going into clinical trials, very few of them actually received FDA approval and progressed to clinical use. And so for context, there have been almost 470 distinct drugs that have culminated in almost 3,000 clinical trials to date. And the ones pictured here are the only ones that have progressed to clinical use with FDA approval as of 2020. But even among the FDA-approved drugs, development can get incredibly slow. And I want to pick out venetoclax as an example. For venetoclax, which is a BCL2 inhibitor, BCL2 was first nominated even as a target in the early 2000s. And it wasn't until 2004 that the first selective BCL2 inhibitor, called ABT-199, started phase 1 trials. It wasn't then until 2011 that ABT-199 started a phase 1 trial for chronic lymphocytic leukemia, and it wasn't until 2013 that there was actual preclinical work that showed a potential in AML. Fast forward to 2016, we saw the first single-arm phase 2 monotherapy trial that had pretty limited but also really promising results. And then all the way from 2016 to 2018, we saw combination phase 1b and phase 2 studies with drugs like azacitidine and other compounds that started to produce really good response rates. And then eventually in 2018, the FDA granted accelerated approval for newly diagnosed AML in patients that were older than 75 years or ineligible for intensive chemo. And then fast forward to, I think, November of 2020, venetoclax finally received full FDA approval for the same indications. So patients that were older than 75 are ineligible for chemo based on phase 3 results that demonstrated an actual survival benefit. And so what we can see here is that resources and funding are poured into the development of AML drugs, but hundreds of them die in clinical trials, and the drugs that do persevere take decades to progress to clinical use. And so the motivation for ATLAS-AML is to see if we can find a way to accelerate the development of promising drugs just like venetoclax, while also improving the chances of survival for other drug candidates all the way from the outset. Now, to facilitate new translational efforts in AML, we have to revisit our approaches for target identification and start by incorporating our existing knowledge of heterogeneity. And I think that heterogeneity in AML has been extensively characterized from at least 3 different perspectives that I'm going to outline. First is genomic heterogeneity. which describes how chromosomal alterations and driver mutations will generate a very clinically heterogeneous group of diseases that all have different drug sensitivities and prognoses. Second, from a functional perspective, we've previously shown in our lab that leukemia stem cells, or LSCs, which have much higher self-renewal compared to a bulk leukemioblast, are actually the cells that are responsible for driving the disease as well as chemoresistance and relapse. Third, more recently in 2022, our lab proposed a cellular hierarchy framework for AML, wherein patient-specific hierarchies with different cell type compositions are actually linked to much different outcomes and treatment response. And so these different aspects of AML heterogeneity all offer very complementary perspectives into AML biology. And so it's important that we consider them together in future drug development and discovery of AML. Now, to refine our understanding of AML heterogeneity, researchers over the past couple of decades have undertaken a lot of transcriptomic studies. And this has generated really large volumes of bulk and single-cell RNA-seq data. But all the subsequent RNA-seq analysis is really technically challenging, and it often hampers a scientist's ability to actually translate insights from all that RNA-seq data into the clinic. Now, there's a couple of transcriptomic tools that have been reported for AML to streamline this process, but they primarily enable gene expression within specific AML subtypes, or they look at associations with clinical characteristics of patient survival. There is no existing tool that will integrate gene expression with functional leukemia stem cell properties, relapse dynamics, and any other non-genetic hallmarks of AML homogeneity. And so this creates a gap that we can see here between wet lab scientists and computational scientists that are working in their own ways, identifying and developing AML drugs. And so we wanted to build ATLAS-AML to bridge this gap and enable a kind of more complete utilization of all the RNA-seq data that spans the genetic as well as the stem cell and functional perspectives of AML heterogeneity. So ATLAS-AML actually integrates 30 bulk and single-cell RNA sequencing data sets with pre-configured pipelines that we've made to help scientists that are working on a new drug answer 3 really fundamental questions that are related to their gene or their gene signature. Question number 1 is exactly which cells express my target? And to answer this question, we collected single-cell RNA sequencing data on 975,220 cells spanning 283 donors to give a really detailed picture of exactly where genes are expressed along the normal and the leukemic hematopoietic hierarchy. And so these analyses can help a researcher determine 2 things. One is if the expression of their gene or their gene signature is higher in leukemia compared to the normal hematopoietic system. And 2, which cells along the whole differentiation trajectory actually express that target the most. The second question I want to help you answer is my target enriched on leukemia stem cells? Our lab has previously developed gold standard leukemia stem cell profiles by performing RNA-seq on LSC engrafting and non-engrafted AML fractions that we evaluated for functional activity by actually doing xenotransplantation. And so this question and this analysis can help researchers determine if the expression is higher on the leukemia stem cells that we know to drive disease progression and relapse. And the third question that we really want to help you answer is which subtypes are enriched for your target. The first 2 questions do a lot of work to help establish the target's actual therapeutic potential, but this question will ask which patient population should we actually prioritize for a potential therapy. So we start by looking at genetic clinical subtypes where we collected RNA sequencing of about 1,080 AML patients and linked the gene expression to genomics and clinical characteristics based on all the clinical data that was available. For clinical outcomes, we compiled overall survival data for 1,008 bulk AML patients. And then for relapsed AML, we actually performed our own RNA sequencing on 29 paired AML samples that are collected at diagnosis and then relapse following induction chemotherapy, and combined our in-house cohort with another 39 diagnosis relapse pairs that we found from 4 publicly available data sets. And then on top of all of this, although ATLAS-AML's primary focus is adult AML, we've also compiled some RNA-seq data and clinical annotations surrounding genomics and patient survival and induction failure for about 300 patients using the target database to allow users to explore their genes in pediatric AML as well. Now, to demonstrate how ATLAS-AML can actually guide target prioritization and characterization, we used ATLAS-AML to benchmark all the published transcriptomic targets that are currently reported for AML. So we performed 4 literature searches on Ovid to find about 368 targets that people were publishing in the literature to be either enriched in AML compared to normal, associated with leukemia stem cells, predictive of poor survival, or overexpressed at relapse compared to diagnosis. And we took all 368 of these targets, we put them on the same playing field, and we systematically analyzed them in ATLAS-AML to see how their performance benchmarked against each individual outcome. And I'm going to walk you guys through exactly how we did that, going back to our simple 3-question framework. And the first thing that we wanted to do was to measure the gene expression across the hierarchical differentiation states in both normal and leukemic blood development. And so in our analyses, among the 368 targets, 81 of them were significant in leukemia cells compared to normal cells. And among them was this gene called Wilms tumor 1, or WT1, which emerged as the top differentially expressed gene in that analysis, which is what's portrayed here on the slide. And so here we put together pseudobulks of almost 600,000 single cells across 11 differentiation states of 168 AML patients and 41 healthy controls. And what we can pretty clearly see here is that across the stem and the progenitor compartment, WT1 was massively overexpressed in leukemia compared to hierarchy-matched healthy controls. And we saw that this was consistent across a number of different genetic subtypes. And so the conclusion from this first question, or this first analysis, is that WT1 might be a gene that we could be able to target while sparing the normal hematopoietic cells. For our next question, we asked whether targets were enriched on leukemia stem cells. And again, this is where we use gold standard profiles of functional leukemia stem cell activity that was validated by xenotransplantation. And in this context, when I say functional LSC activity, I mean that LSCs are functionally defined by their ability to engraft in immunodeficient mice and actually generate leukemic repopulation. And so among our 368 targets, we saw that 38 of them were associated with LSC engraftment, and 5 of those overlapped with the 81 genes that were enriched in AML compared to normal cells. And again, we saw that WT1 emerged as one of our top candidates and was highly enriched in the LSC engrafting samples compared to those that didn't engraft. But we also know that leukemia stem cells can be pretty phenotypically diverse. So we also looked at WT1 expression across the immunophenotypic subpopulations that were sorted by CD34 and CD38. And we saw that WT1 was much higher in the CD34 positive, CD38 negative, and the CD34 positive, CD38 positive compartment, which we know is strongly correlated to leukemia-initiating cells. And so the conclusion from this analysis, or this question, is that targeting WT1 could be a promising avenue to disrupt the cells that are actually initiating the leukemia within AML. On to our third question, these first 2 analyses validated that target enrichment on the leukemia stem cells might indicate some sort of role in leukemogenesis, but the actual translational impact of these targets is going to depend on which patient populations express it. And so to address this, we looked at target expression across 3 key dimensions of patient stratification. The first was high-risk genomic and clinical profiles. The second was overall patient survival outcomes. And third was disease progression from diagnosis to relapse. These criteria all represent different methods of looking at high-risk patients. And so we said that meeting any one of them would be sufficient to warrant some sort of future consideration for therapy. So to begin, we wanted to characterize the translational potential at diagnosis. And so we looked at target expression across ELN, or European Leukemia Net Risk Classification System, and our cohort of bulk of 1,079 patients. And so the ELN system accounts for all sorts of cytogenetic and mutational and clinical characteristics. And what we saw was that 129 of our original 368 targets and 2 of the previously 5 benchmark targets were associated with high-risk ELN profiles. But as you can see here, for WT1, we actually saw that expression was associated with a more favorable or intermediate ELN category at diagnosis compared to the high-risk adverse ELN patients. Now, for the next subtype that we wanted to look at, which was clinical outcomes, we linked gene expression from about 1,008 patients to overall patient outcomes. And what we saw was that 26 of the 368 reported targets and 1 out of the 5 previously benchmarked targets was linked to a poor prognosis. And for W21, we saw that across individual cohorts, as well as here in a pooled multivariate Cox regression that we stratified by three patient cohorts, W21 wasn't predictive of patient survival. And it's also important to note that this prognostic value, it stayed insignificant even when we adjusted the multivariate analysis for different clinically relevant variables like age and white blood cell count and FLT3-ITD status and cytogenetic risk and so on. But thirdly, although a lot of targets are initially looked at or identified based off expression at diagnosis, we know that a lot of experimental drugs are first tested on patients who have relapsed or are refractory to chemotherapy, which is why we included relapse as a third kind of subtype of patients that we're going to look at, and also why we decided to perform RNA-seq on 29 paired samples to be able to interrogate relapse at a much larger scale. And what we saw was that 25 of our original 368 targets were upregulated at relapse compared to paired diagnosis samples, and that included all 5 of the benchmark targets. And as you can see here, WT1 was the one that was most overexpressed at relapse compared to diagnosis. And so what we would want someone to take away from this analysis is that rather than targeting WT1 at diagnosis, where it wasn't strongly associated with adverse clinical or genomic risk, an anti-WT1 therapy might be better prioritized for relapsed AML patients where its expression becomes much higher, and we also know where treatment options are particularly lacking. Now, as we mentioned before, although our primary focus is in adult AML, ATLAS-AML also has some genomics and treatment response and survival data within pediatric AML. And we look at this to see if we can find a similar expression pattern in the pediatric side of the disease as well. And what we found was that, although WT1 wasn't highly associated with any of the genomics or survival parameters that I looked at, what we did see, which is pictured here, is that WT1 was upregulated in patients that had an MRD, which is minimal residual disease positivity after 1 course and 2 courses of induction chemotherapy. So, perhaps here we could say that persistent WT1 expression might contribute to like chemoresistance or poor clearance of leukemia cells in pediatric AML. But this is just meant to give a bit of an exploration of the pediatric side of things as well, and say that, an anti-WT1 therapy that gets developed might warrant some exploration in pediatric patients as well. So altogether, we built ATLAS-AML to basically give people like a one-stop shop for looking at transcriptomic data that pertains to normal and leukemic hematopoietic hierarchies, functional leukemia stem cell activity, genomics, clinical outcomes, and relapse as well. And to make sure that this pipeline is accessible, we streamlined ATLAS-AML into one R package that requires really minimal bioinformatics dollars to be able to use. But in addition to this, I want to leave you guys with some takeaways on how to interpret and use ATLAS-AML for your drug development research. The first is we think that ATLAS-AML can be particularly useful in preclinical drug development for target prioritization. We've worked with a lot of people that do really cool high throughput screening methods like CRISPR dropout screens or we do bio-ID, and these experiments generate really extensive lists of gene candidates that they then have to find a way to go through to pick something out to pursue. And so benchmarking these gene candidate lists in ATLAS-AML, exactly how we benchmark the published targets in ATLAS-AML might represent a way that we could speed up the identification of more promising candidates before we actually commit to experimental validation, which we all know is really time and resource intensive. Another observation I wanted to point out is that we saw that several of our published targets that were reported in the literature, which we analyzed, they're often optimized for very particular features of AML heterogeneity. So for example, we saw a lot of targets that were very enriched on the LSC and grafting population, but in nothing else, or a lot of genes that were very strongly associated with poor patient survival, but nothing else. And it was pretty rare that we saw a couple of genes that actually coalesced on all 3 perspectives. And in fact, WT1 out of the 368 was one of the only ones that did that. But what we want to emphasize with ATLAS-AML here is that having therapeutic potential is not confined to being enriched in all of the different analyses or all of the different perspectives of AML heterogeneity. Instead, it's important that the researchers using ATLAS-AML consider what analyses are going to be relevant to their targeting modality. And what I mean by that is if you take immunotherapy targets, for example, genes that are expressed on like the normal hematopoietic cells may not be suitable for a CAR T therapy where the target expressing cells are all kind of indiscriminately eliminated. But if a researcher's targeting approach was using like a small molecule or an siRNA approach, then an expression in normal hematopoietic system might be less concerning if that drug is targeting like a specific functional dependency of AML as opposed to absolute expression. And so instead of trying to enforce a rigid selection criteria for targets, we want ATLAS-AML to just give a structured framework for preclinical decision making to help researchers be able to contextualize the target expression patterns that they see in relation to what their targeting approach is. Yeah, that concludes my presentation on ATLAS-AML. Thanks so much to the T-CAIREM team for giving me the opportunity to present my work. And I, of course, want to thank my mentors, John Dick and Andy Zhang, who supervised this project. And I also want to thank all of our collaborators around the world who are currently using ATLAS-AML in their active projects. Thank you.
Great job, Suraj. Congratulations. That was a great body of work and really nicely presented. I'll maybe open it up first to the co-adjudicators if you want to start with questions. Otherwise, I can go ahead.
Sure, I'll ask a question. Yeah, thank you very much for the presentation. Very interesting work. So you mentioned a few times that one of the issues with this field or with this topic of research is the heterogeneity present in the disorder and the targets and the treatment. And then you took a sample and basically said, oh, these are the cases that we are familiar with or that we can work with, and that's what you built your package on and what you can compare your findings with. So I'm wondering, what is your impression of the representativeness or the size of your sample here compared to the heterogeneity out there in the world? Is there some bias there? Is there some cases that are missed? How much? I mean, of course, you have more data, but can you give us an impression of that?
Yeah, that's a great question. I mean, of course, like heterogeneity in acute myeloid leukemia is looked at from a lot of different perspectives, from different labs around the world. I find that when we, like, at least in our lab, the way that we look at heterogeneity, especially being a stem cell lab, as a lot of our work has demonstrated that's like the leukemia stem cells and its primitive leukemia hierarchies that can drive a lot of the disease. And so for us, that was kind of the unique angle that we wanted to bring into what a lot of the existing tools don't have. So there are tools out there right now like Bloodspot or LASA or Visome that have adopted kind of the more traditional genetic or clinical look of heterogeneity in AML, where they already enable people to take their gene expression or take their gene signatures and see how they relate to patient survival or see how their expression differs within different genetically defined subtypes. But there's no tools out there right now that allow people to look at how it relates to functional leukemia stem cell activity, that allows people to see how it differs at relapse compared to diagnosis. And so when we were thinking about, what perspectives of heterogeneity are key to the biology, but missing from a lot of these tools, that's what we landed on. And so I find that, integrating that with the genetic perspective, of course, it doesn't capture everything, and that's probably a model that would need to be updated in the future. But I think that brings in a very key other dimension of the disease.
Dr. Mitsakakis.
Yes, hi, Suraj. Thank you for the interesting presentation. I have one comment to say that actually my work is quite different from this area of research. I had a hard time to follow very closely to what you're saying, but one thing I noticed and I was thinking about is essentially the relationship of your work with AI. So as far as I understood, and I could be wrong, the methods that you use for the analysis is very simple, mainly descriptive, and occasionally some tests to compare different groups. Am I missing something? Is that how do you see the relationship of what you've done with artificial intelligence? And a second part of this question is that maybe you haven't done this yet, and in which case, how do you see to extend what you have done with the use of AI?
Yeah, it's a great question. I'll actually go back. Slide. So a lot of where AI played into our project is we collected a lot of data pertaining to the normal and the malignant hematopoietic hierarchies. And then we used a lot of like unsupervised cell clustering to annotate these differentiation stages across both the normal and the malignant hierarchies. So a lot of the AI portion of the project comes from doing unsupervised cell clustering to annotate the cells across the normal and the malignant hierarchy. We have some other data sets, for example, this is entirely in the bone marrow, but we have another normal hematopoietic data set with cord blood that is included in the pipeline, but not pictured here, where we'll also use cell clustering to annotate the different cell types across the data set. So that would be the main application of is unsupervised cell clustering for the hierarchy information.
Okay, great. Thank you.
Suraj, I was just wondering, like this sort of pipeline of multi-omic data integration and bioinformatics, like, it's so common across so many different cancer types. Do you see generalizing this sort of package and kind of final product across other cancer types?
Yeah, really exciting question that you're bringing that up, actually, because some of my other recent work has been taking a very similar concept to this and applying it to other types of acute leukemias or other myeloproliferative disorders. The main inspiration for this project basically came from knowing that there was a lot of transcriptomic data that was out there, but a lot of experimental scientists that I've worked with have a hard time kind of harnessing that data and using it as its full potential. And I think that same issue exists for any sort of cancer disease in general where large volumes of data exist. And so one example of where we've started to expand ATLAS-AML to other diseases is with myelofibrosis. Our lab has a cohort of around 400 patients at Princess Margaret that we recently performed RNA sequencing data on in myelofibrosis, and that represents one of the bigger RNA-seq cohorts that's out there. And so in a very similar fashion, I've taken kind of all the clinical and the genomics data and linked it to the gene expression and built a similar kind of pipeline within myelofibrosis that allows people to really quickly look at all the associations between gene expression and clinical outcomes and genomics and so forth. So I think it's a great question. I think these types of models are ripe to be applied to other diseases, and I think they're coming soon.
Awesome. Congratulations. Great presentation. I think because we have time on our side and I don't see questions in the group chat, we'll maybe switch over to the second presenter. So just briefly introduce Sidrah. Sidrah Laldin is a first-year cardiac surgery resident at the University of Manitoba. Since her medical school training at McGill University, she's been interested in researching and developing consumer-centered, data-driven applications in healthcare using design thinking methodologies. She's particularly interested in applying artificial intelligence-based methods to improve surgical outcomes for cardiac surgery patients. We'll pass it over to Sidrah. Once again, 20-minute presentation and 10 minutes for questions.
Okay, thank you for that warm welcome. Just give me a moment. Share. Can you see my slides?
Yes. Right now, just in the...
Oh, sorry.
Yeah, perfect. Awesome.
Okay. Welcome everyone to this talk. So I am talking about comparing innovative artificial intelligence algorithms to assess echocardiographic videos for clinical modeling. As mentioned, my name is Sidrah Laldin. I'm a first-year cardiac surgery resident at the University of Manitoba. This project was sort of a multidisciplinary teamwork consisting of biomedical engineers at McGill University, where I did my medical school, by Cedric Shumtim, who was a biomedical candidate in PhD, Dr. Satya Prakash, who's a biomedical engineering professor at McGill University, and Dr. Dominic Shumtim, who's a senior cardiac surgeon at McGill University. So in terms of cardiovascular imaging, the expansion of machine learning algorithms in cardiovascular imaging has sort of created this phenomenon where we can look at repositories of hidden information within imaging with different imaging modalities. one of the main imaging modalities, sort of staple in cardiovascular surgery and cardiology, is echocardiography, or echo. It's an ultrasound-based modality to assess cardiac function and cardiac pathologies. There are two kinds of echoes. Just for completeness, I'll mention them. One is transesophageal echo, which is used intraoperatively to assess cardiac function and hemodynamic stability while the person is going through cardiac surgery or sometimes non-cardiac surgery. The other one, which we focus on our study, is transthoracic echo, which is used as a screening tool to assess for cardiac function and as part of the initial disease management and risk stratification in both medical and surgical decision-making. Now, as widely as echocardiography is used, there's a huge level, and it's documented in the literature, high level of inter-observer and intra-observer variation on how different parameters are measured. And in this context, artificial techniques, including machine learning, can offer potential benefits for getting more precise, more reproducible parameters and outcomes. So here is what I showed with you. The last slide had still images. This is how echocardiography, when we look at echocardiographs in the labs or in the hospital, this is what we're looking at. We're looking at video images. We're looking at heart function, chamber function. We're looking at how the valves are functioning. And this is what we look at. It's a video sort of based inherent modality. So some of the gaps in the literature is, so far the research, although it's changing now more so, is on echocardiograms at least, is research based on still image-based modalities, which kind of takes away the information that can be gleaned from looking at both spatial and temporal aspects of echocardiograms. And these gaps are due to scarcity of available video data sets that are well annotated data. And if you look at the literature, what you will see is people have done, they're doing research on this, and there's a lot of heterogeneous data sets, and there are various machine learning algorithms that are used. And which is good because, we're experimenting, we're showing this versioning of different tools and algorithms being used, but it often leads to lack of standardized comparative assessment of performance across different models. So, one of the things which I will talk about later on is we have standardized data sets that have been sort of published recently in the last couple of years, which we use for our comparison on this experiment. And these standardized data sets offer performance benchmarks to better evaluate and compare different model architectures. And this allows for quality assessment, allows for applicability assessment before adoption in clinical practice, which I think is really important before they are adopted. These machine learning models are adopted in clinical practice. So you need robust quality assessments. And one of the metrics, as kind of mentioned in our last talk, was the size of data sets which are used to train algorithms is very important and has a strong impact on design and performance of machine learning models. And this is really relevant across all machine learning fields, but particularly relevant in medical imaging, given lack of high quality, well annotated data. So to address this, a couple of years ago, Stanford University published sort of a publicly available cardiac motion video data set called EchoNet Dynamic, which had over 10,000 echocardiogram videos which were annotated. And this was curated to predict left ventricular ejection fraction. I'm not going to get into the science of it too much, but just to let you know, the left ventricular ejection fraction is the most studied echocardiographic parameter of left ventricular systolic function because it has implications for goal-directed medical and surgical therapies, and it's used a lot making surgical and medical decisions. So just to talk a little bit about some of the learning paradigms that were used in our study, and most of you are probably familiar with it, but this is from a clinician's point of view that I'm addressing these. Learning paradigms refer to approaches and frameworks that use to train predictive models We define how data is utilized. They define what types of feedback is provided during the learning process and how model performance is evaluated. So primary learning paradigms are supervised, unsupervised, and reinforcement learning. And in between them, you have, these are the three primary ones, and you have others, which are semi-supervised, few-shot learning, meta-learning, self-supervised learnings. And these paradigms play a crucial role in creation of intelligent systems. They have their set of advantages and disadvantages and challenges. So we focused our study on supervised learning and semi-supervised learning to see how each of these models perform with small amount of data sets. So for supervised learning, these are algorithms that are trained on labeled data. These are input-output pairs where input is given as a data to learn and output is a correct associated with the input. You learn mappings from input to output. The algorithm learns these mappings so it can accurately predict output for new unseen data. So on the chart here, right, I have labeled data. I give it labels. We do machine learning models, creates a machine learning model. We give it test data and makes the appropriate predictions. And it minimizes the difference between models. The way it does the learning is minimize difference between models predictions and actual labels by adjusting parameters like gradient descent. And it's used in tasks like classification and regression. And from the literature, when you read it, it seems like this is the gold standard in medical imaging at the moment, using supervised learning models. The semi-supervised, on the other hand, bridge the gap between supervised and unsupervised by leveraging both labeled and unlabeled data. Obtaining large amount of labeled data is quite extensive, is time consuming. And one of the ways, there are different algorithms for semi-supervised learning, but one of the algorithms is self-training, which I mentioned earlier. It uses a small set of labeled data to guide the learning process while also extracting useful information from larger pool of unlabeled data. So over here, you have labeled data. You train the model with it. Then you give it unlabeled data. It presents what's called pseudo-labeled data and predictions. And then you add that with labeled data. And that basically presents a model, a learning model for us. And the strength of this algorithm is that it takes both labeled data and unlabeled data to achieve higher accuracy and performance compared to purely supervised and unsupervised models. And it makes it where domains where labeled data is quite scarce. So in this study, we examined self-training for semi-supervised performance. So our research hypothesis was that although as I mentioned, supervised data is a gold standard, we put forth that we argue the lack of clean labeled data in TTE settings suggests that a semi-supervised learning may be more beneficial in deep learning applications. And this is because it may obviate the need for large sample sizes for determining sound, clinically valid performance benchmarks. And our experiment objective was, and it was basically a quality assessment experiment, is to use multiple dynamic deep learning algorithms on video-based echocardiographic images using sample size determination as a key variable to assess performance, optimal performance. And we do this by doing a longitudinal analysis on a beat by base to get both the temporal and spatial assessments of using variable data set sizes. And for the methodology, I'll go briefly into this, the algorithms. I'll go into the methodology a bit more later, but the study compares 3D CNNs, video vision transformers, and hybrid convolution neural networks and long short-term memory models, all within the domain of supervised learning and semi-supervised learning. And we basically assess their performance. To kind of give a sense of 3D CNNs, they have emerged as a powerful tool in tasks with video. Unlike the traditional 2D dimensional CNNs that operate just on individual frames, which are images, 3D CNN extend the convolution operation network into the temporal dimension and allow capturing of both spatial and temporal features. And this makes them very effective for understanding dynamic scenes. So over here you have, input into the input frames, you do 2D convolutions, then you do 1D convolution, temporal convolution, and you get a 3D network that gives you both spatial and temporal sort of information about the scene. And this factorization allows the model to learn spatial and temporal features in two separate steps which also reduces the computational complexity and enhances the ability of the network to capture intricate spatial patterns, spatial temporal patterns. Then we have video vision transformers. These are cutting-edge approach to processing video data, which leverage the power of transformer architecture to capture temporal and spatial information. Transformers were actually developed for natural language processing, and they've kind of revolutionized machine learning by using what's known as self-attention mechanisms to model long-range dependencies. Now, self-attention mechanisms, they allow a model to weigh importance of different elements in a sequence and then focus on the most relevant part when they are learning. So video vision transformers extend this concept to applying attention to both spatial and temporal dimensions. We use video SWIN transformer, which is an extension of the SWIN transformer architecture designed specifically for video analysis by adapting its hierarchical structure and shifted video-based self-attention mechanism to capture the spatial and temporal features in the video data. And finally, we have what is known as hybrid models, and they have gained quite prominence in their ability to effectively analyze video data. So they leverage both the CNNs and RNNs, which are recurrent neural networks, to handle spatial and temporal complexities. RNN is a special type of neural network. For those of you who don't know, it's kind of designed to process sequential data where the order of elements is very important. So CNN, kind of the convolution neural networks are really good at extracting spatial information from individual frames, whereas RNNs, such as the LSTMs are part of RNNs, they excel at capturing temporal dependencies across sequences of frames. And this kind of benefits both like a synergistic information sort of interaction between spatial features and temporal features. And the hybrid model that we used in this study is CNN plus LSTM, in which each frame of the input is fed to a ResNet convolution network, which in turn feeds it output to LSTM. So over here you see images. It's feeding the CNN, and the CNN feeds the LSTM, and then you get the output. So our method was, we had 10,000 available, like readily available videos, annotated videos for us that had LVF values from the annotations. So we took the raw data, we pre-processed the data, we segregated the data into labeled and unlabeled data. We made supervised models based on labeled data, and these are the three models that we used, as I mentioned. These are the three algorithms that I explained. And then we use the unlabeled and use the label to do semi-supervised. And we used three models, same models that we use for supervised. And we divided them into sample sizes of 200, 400, and 800 for supervised. And the sample size in semi-supervised, we divided into 100 labeled, 100 unlabeled, 200 labeled, 200 unlabeled, so on and so forth. And we determined the ejection fraction and comparison with annotation with the ground truth. And the metrics we used is the mean absolute error and root mean square error, which are widely used metrics for evaluating performance of regression models. Mean absolute error is the average difference between predicted and the ground truth of LVF values, where a lower mean absolute value indicates average sort of predictions that are closer to the truth value. I'm not going to get into root mean square error, but just for completeness, it emphasizes larger errors more than mean absolute error, owing to the squaring of differences before averaging. And the low root mean square error indicates fewer larger errors in prediction. So what we get is consistency of the model in the long run. So these are the results we found. For supervised learning, mean absolute error, none of this is a surprise. This is, we kind of expected this, where I want to draw your attention that if you, to the x-axis here is, you supervise is on the x-axis, you have 200, 400, 800 with the data size of the data, okay? And then mean absolute error is the on the y-axis. And what you see and what we predict is that as the size goes, increases, you find that a mean absolute error decreases. And this is kind of true in most, in most cases. And what you also see is the hybrid model does perform the worst in terms of having higher mean absolute error. And the ResNet is about, you can't really see it, but the values actually do decrease with increased label data sets. Okay. But the interesting part comes in, this is a kind of busy slide, so I'm going to orient you to the graph here, is when you compare supervised and semi-supervised models. So on the x-axis here, you have supervised, semi-supervised with different label size, different data sets. So 200, 100 plus 100 for semi-supervised, 400 for supervised here, and 200 plus 200 plus for labeled and unlabeled data for semi-supervised. So I want you to take three things out of this slide. And the three things are similar to supervised. When you see the data, as the data set increases, the MAE, the mean absolute error, decreases even for unsupervised data. That kind of makes sense. But what was interesting for us is, and we kind of predicted this because, we were sort of testing our hypothesis, is when you have supervised and semi-supervised with a similar amount of data sets, 200 for supervised and semi-supervised, you have 100 labeled and 100 unlabeled, their performance of semi-supervised in, for example, hybrid ResNet LSTM is actually better than the supervised models. So what we are showing is semi-supervised that have unlabeled data are actually doing better than supervised data in most data sets across the spectrum. On the other hand, the third thing I wanted you to take from this slide is if you look at ResNet 3D, which is a 3D CNN model, the supervised and semi-supervised are about the same. Like there's not much change. There's some change here, but not much change. So what you're seeing is they're performing just as well, if not better, if not less worse, but just as well as the supervised learning models. So our results discussion points is that our results indicate the supervised learning benefits more from labeled data, not a surprise. Our results also show that in some models, the addition of unlabeled data in semi-supervised models significantly increase the prediction accuracy of the models with increasing sample size. And the performance was generally, in some models, were generally comparable and similar across supervised and semi-supervised learning models. And this can be especially useful that semi-supervised models can leverage unlabeled data to bridge the gap between performance when labeled data are inaccessible. And using mixture of unlabeled and labeled data is comparable to the models using only labeled data. So what we show is that in cases of limited data sets, model performance can be enhanced with the addition of unlabeled data sets, obviating the need for large samples of labeled data. There are some limitations for our study. Findings are very data set specific. This is the strength of the study too, but it's also the limitation of the study. It's very data set specific. We're only focusing on an EchoNet dynamic. So these are not generalizable results. Obviously, we're not arguing for that to other echocardiograph data sets and tasks beyond LVEF prediction. The models tested, although they're popular for video analysis, do not cover the full range of semi-supervised optimized models, which could produce different results. And then we only examined some very basic semi-supervised learning algorithms in self-training, leaving room to explore other methods that might produce enhanced performance. When we submitted our paper to Journal of Thoracic Cardiovascular Surgery, they said, we want to know what the training times are, because this is kind of interesting if this is being adopted in clinical settings, what the training times were and testing times were. So we did a bit of study on that. This was a side thing. But what we found is CNNs, the 3D CNNs, were cheaper than the sequential processing of LSTMs, which obviously because it's sequential, they're more expensive, computationally more expensive, or the attention mechanism in transformers. The LSTM's current step depends on the previous one, which makes them less parallelizable. So you can see they're quite, they're quite expensive. And the transformers have a high computational cost due to self-attention mechanism, which requires computing pairwise interactions. So conclusion, what we are saying is semi-supervised learning models can have comparable or even superior performance to supervised models requiring fewer labeled data sets, which helps overcome the clean data scarcity in computational medicine. And these findings contributes to advancing deep learning techniques in the field of medicine by improving model efficiency and accuracy. And future studies should explore diverse data sets, novel supervised, semi-supervised methods, and robust model architectures to enhance generalizability and clinical applicability of these approaches. Thank you very much.
Congratulations, Sidrah. Great job. Very nice presentation. Dr. Voppel, I saw your hand go up, so I'll pass it over to you.
Yeah, thank you very much for the presentation as well. Interesting topic. So I've worked a bit with convolutional neural networks, and one of the things standing in the way of clinical implementation of that is usually the explainability of the model. So what is actually happening behind in the black box of the model, rather than just the outcome, correct or incorrect, or in this case, an estimate of the flow there. So I was wondering, these video models that you use, can you also dive into the model and see what they're actually looking at, or what data in the model over time they're using to predict their prediction?
I'm sure I know that we didn't do this. It was I was involved with the clinical aspects of it. Our team, biomedical engineering team, was involved with more the programming side of it. But I understand, I've read in the literature that there are different ways you can create heat maps. You can create like weights on how much a certain aspect is contributing to the weight of the, contributing to a certain weight of the result. We did not do this, but I am sure I have read in the literature that it is quite possible, and there are newer research that is actually exploring that, especially using what they call heat maps in the literature, which is gaining robust attention in that field.
Yeah, well, I think it would be very interesting to compare that to the expert human raters, to say like, oh, we look at this ventricle and we see it moving back and forth, or it becomes less or more intense at a certain point, and you would see if the model sees the same thing. And it might also give an idea of where the errors are coming from. Thank you.
Thank you.
Dr. Mitsakakis, go ahead.
Yes, thanks, Sidrah, for the excellent presentation. Just I have a few questions, but just focus on only one or two. I was just curious. It seems from what I saw that you only used one data set per sample size, which I find I would advise not to do that, because you never know whether that was just one-time thing. I mean, in order to obtain more robust results, advice would be to try different samples from the original data set, and see if the performance changes. And one other thing I wasn't sure is, how do you obtain the performance measures? Did you use cross-validation or something? What's exactly the error that you show? What exactly is that?
Yeah, I think my biomedical engineering team would be more like adept at answering that question. From what I understand is we had 10,000 data sets, and we usually basically use those, subdivided them, use them as a validation data set and testing data sets. So of those, we sampled 100, 200, whatever, whatever the algorithm allowed for. That is my understanding of what they did.
Let's see. And then one comment to say, this is, I guess, it's an interpretation thing. You mentioned that you found a significant and improvement in the performance when we move from, let's say, 400 to 800. But the way I saw it, with the exception of the third method that it was the worst, the other things actually I found the improvement very minimal. So I guess it's a matter of interpretation,
We can go back to the chart here. So you're talking about this.
Yeah, I don't really see a big difference between 400 and 800. I mean, with the exception of the green bar, which I can see is big difference, but the other two are almost the same, right?
Yeah. I mean, that's you're right in that, and I can revisit. I mean, when we did that, I don't have the numbers exactly with me, but when we do the numbers, maybe they don't show on the chart, but the numbers did show less. But you are right, and that's the other thing we're arguing is that, you know, because of lack of clean data, they may, you know, we're basically, our interest is, as much as that we're comparing data sizes across the same supervised learning models or in semi-supervised learning models, we are basically wanting to compare between supervised and semi-supervised. And the main takeaway from us, for us, was that there should be, although supervised is considered the gold standard, we should entertain semi-supervised because they are performing comparably to the semi-supervised. You know,
No,
they're not worse.
I agree with you. I mean, this is very interesting. I agree with you. Yep. Great. Thank you.
Thank you.
Sidrah, maybe I'll ask a question just a little bit more on the clinical side. So I'm sure you might have seen or maybe not seen, I think it was only published 2 days ago, but JAMA just put out a pretty big paper with basically deep learning based like assessment of echocardiographs across, I think, 39 different measurement indices. The future of echocardiography, do you think it's going to be humans doing it, or do you think we're going to move to a point where it'll be fully automated, like our echo fellowship's done?
I think there's obviously it's never going to be a complete takeover, but that's a very interesting question. And when I sit with the graphers, you know, we actually hand manually segment them, and these are the tasks I think can be really taken over by artificial intelligence. But then you have pathologies, you have noting different kinds of pathologies. What kind of valvular pathologies are you seeing a thrombosis? And these are things that clinical assessment will still be for, I think, in the hands of clinicians. But there is definitely a large sort of threshold, a large kind of way where artificial intelligence can definitely, from the current status that it is now, can use to improve practice. Because right now it's basically you're pointing on you go on the software and you click on certain points and you tell the system, oh, this is the structure of the left ventricle. And these are things I feel can be automated, and there's definitely room for, you know, automated assessment. But I think at the end of the day, how do you interpret those results? What do those results mean? What is the, you know, you can measure valvular sort of function by certain numbers, but then there are other things that are clinically happening within a particular valve or a chamber that I think will, you know, rest in the interpretation of the clinician.
Perfect. Thanks for that answer. So it sounds like might shift workflow, but maybe not necessarily fully obviate certain things. But very interesting work. Thanks so much for presenting it at the T-CAIREM rounds. Just want to thank everyone who attended. Thank you also to the adjudicators and to Marianne, as well as the two great presenters. That concludes the rounds for now. We'll hopefully, oh, actually, I just saw something pop up. Okay, I can maybe answer that question. I think that was directed at just me. So thanks very much, everyone. We'll see you next time. Have a great rest of the week.